bitin. Sa manlulupig, di ka pasisihil. Sa dagat at bundok, sa simoy at sa langit mong bagaw. May dilagang tula at awit sa paglayang minamahal. Ang kisap ng watawat mo'y tagumpay na nagdiningning. Ang bituin at araw niya kailan pa may di magdidilim. Lupa ng araw ng buwan, ating pagsinta, buhay ay langit sa piling mo. Aming ligaya ng patay mga api, ang mamatay ng dahil sa'yo.
In its 44 years of existence, NCPC is at the forefront in providing solutions to the country's crop pest problems by developing, adapting and extending appropriate pest management technologies to farmers and formulating appropriate recommendations for use by government policymakers. Our vision: world-class excellence in crop protection research, extension, and policy support programs to ensure food security, environmental sustainability and upliftment of the general public's quality of life. Our mission: to train competent scientists and professionals with passion for excellence in science, global in perspective, and committed to the service of the country. To develop environmentally sound, sustainable and cost-effective pest management programs and technologies to address the crop protection concerns of growers and farm holders. To disseminate scientific knowledge and provide technical assistance in pest management. The center is headed by a director, deputy director and division heads supported by competent staff composed of scientists conferred by DOST and CSC, senior and junior researchers, and administrative staff. The center's agriculture programs are focused in plant biosecurity, plant pest management, pesticide management, public service, and advisory services. The center provides technical services to the public through its plant health clinic by providing plant health diagnosis, pest and natural enemy identification, and pest management recommendations. The center is also proactive in addressing serious pest problems through quick response to pest outbreaks. The center hubs newly equipped laboratories with cutting-edge equipment to support researches in the fields of entomology, plant pathology, pesticide management, vertebrate pest, and weed science. The center generates knowledge through extensive research and deep extension programs that translate into public service. It provides international and local training to disseminate research findings and crop protection technologies. The center reaches out to the general public through publications, visitor services and promotional activities through showcases and digital media. NCPC addresses food security and food safety concerns through promotion and continuous development of technologies. Through the years, the center have identified several biological control agents against major pests. NCPC continues to excel in the fields of crop protection and pest management, innovating endlessly to attain world-class excellence on research and public service.
Okay, good morning everyone. Narito po tayo sa third episode ng ating anniversary webinar series. Ito ay nihahandog ng National Crop Protection Center for its 45th anniversary with SPM, Reshaping Pest Management, Responding to Challenging Signs through Relevant and Unwavering Research and Extension. Ako po si Mary Joy Mendoza, ang inyong magiging host for the day. Just to give you a recap of the previous topics ng ating webinar series, ang unang episode ay ang back-to-back talk patungkol sa fall armyworm. Ang bago ang makuminsalang harabas ng mais ni scientist Mario Navacero. At ang isa pang topic ay ang halaman at pangangasiwa ng harabas ng sibuyas ni scientist Marcela Navacero. And for the second episode, last Wednesday, we had an interesting topic on mango pest delivered by Miss Jenna Joy Apolinario. And for our third episode, we will be having another exciting topic. But before that, let's have a reminder first for today's webinar. Para sa ating house rules, ang webinar po na ito ay matutunghayan ninyo sa tatlong platforms. Dito po sa Zoom, live din po tayo sa NCPC YouTube at sa NCPC Facebook page. Kung mayroon po tayong mga katanungan habang nagde-discuss ng ating speaker, huwag pong mag-asibiling ilagay ang inyong mga tanong sa Q&A box na makikita ninyo sa Zoom o di kaya naman ay sa chat box natin sa YouTube at Facebook. Atin pong imo-monitor ang inyong mga katanungan at sisikapin po natin iparating sa ating speaker sa Q&A portion. Okay, malinaw at maayos po ba na nakikita o naririnig kung saan po man kayo sumusubaybay? Atin pong alamin ng mga lugar kung saan po kayo nanonood. Buksan po natin ang ating mga browser at i-type ang menti.com na pina-flash po sa screen ngayon at i-type ang code na, hintayin lang po natin. Muli, buksan po natin ang ating browser at i-type ang menti.com at i-type po ang code na, saglit lang po, hintayin lang po natin muli. Yes, yan. At i-type po natin ang code na 5299-2197. Sige po, hintayin po natin ang ating mga participants. We would like to know your current location watching this webinar episode. Kindly type menti.com in your browser and use the code 5299-2197. And then put your location, province if you're here in the Philippines, or your country if you're watching from abroad. Okay, hintayin lang po natin makapasok ng ating mga attendees. Magkano po kayo? Matagal po kayo mag-graduate dito? Kasi daw po, meron po kong webinar. Okay, mukhang nagkakaproblema po tayo. Siguro po ay i-chat po natin sa chat box na lamang po kung saan po tayo nanonood. Okay po. Magandang umaga po sa mga taga-Bae, Laguna. Meron din po tayong mga participants from PSAU, Magalang, Pampanga. Sa CLSU, PhilSCAT. Good morning po sa atin. from Benguet. Meron din po tayo sa Nueva Vizcaya, Zamboanga del Norte. Okay, ang dami po natin From Davao del Norte sa Panabo City. Good morning po. From Cebu, sa Calawag, Quezon. Good morning. Sa BSU, Benguet State University. Meron din po tayong participants from CLSU, Philscat. From La Union, good morning po. Sa Apayo State College. Echage, Sabela, sa ISU, good morning po. Meron din po sa Davao City. Magandang umaga po sa ating lahat. Good morning from Paumbong, Bulacan. And we would like to acknowledge then yung presence ni Mr. Stephen Arthur from Ghana. Wow, we're going international. Good morning po from Lucban, Quezon. Good morning po sa ating lahat. Okay. So, may hahabol pa po ba? Maraming salamat po. Okay, parami po tayo ng parami. Nako, marami na po ang naaabot ng ating webinar. Maraming salamat po sa inyong pagdalo sa ating webinar episode na ito. Okay po, to formally start our program, tunghayan po natin ang paunang mensahe ng buting direktor ng National Crop Protection Center, Director Melvin D. Ebuenga. Sir Melvin?
Sa ngalan po ng CAP Extension Committee at ng National Crop Protection Center, welcome po kayong lahat sa webinar na ito. Una sa lahat, nais ko pong pasalamatan si Dean Agbisit at ang CAPS Extension Committee para maisakatuparan ang gawaing ito at syempre ang mga kasapi ng NCPC Seminar Committee at Public Service Division sa pagsasaayos para maidaos ang webinar na ito bilang bahagi ng mga palatuntunan sa paggunita ng 45th NCPC Anniversary. Maraming salamat din sa ating resource person na si Mitch Guerrero na kahit na nasa New Zealand at nag-aaral sa kanyang PhD degree, ay pinaunlakan ang aming paanyaya na maging speaker ngayong araw. Mukhang marami tayong matututunan sa ating topic na The Hidden Wonders of the Soil, Nature's Solution to Pest Management. Nasa lupa lang pala matatagpuan ang solusyon sa pagsugpo ng ating mga peste sa halaman. Abangan na lang ninyo mamaya ang mga detalye kung ano-ano ang mga bagong biocontrol agents o microorganisms at paano natin sila magagamit sa IPM. Inaasahan namin marami kayong bagong mga kaalaman na matututunan pagkatapos ng webinar na ito. Hindi man tayo magkita ng personal, umaasa kami manatili ang ating ugnayan at bukas ang NCPC para makatulong sa mga suliranin ninyo sa mga peste at sakit ng halaman. Sa ating lahat, mag-ingat tayo palagi sa pandemic na ito. Maraming salamat at mabuhay.
Thank you very much, Director Melvin. And now we'll proceed to you to introduce our speaker for today's webinar. Our speaker is a university researcher at the National Crop Protection Center and currently on a study leave. She is a PhD graduate student at Massey University, New Zealand. She is an entomologist with expertise in insect pathology, molecular entomology, and crop protection entomology. She is a member of several international and national societies, including Entomological Society of New Zealand, Entomological Society of America, and Philippine Association of Entomologists. She is also a recipient of various awards. To name a few, she is a recipient of the Netherlands Fellowship Program or the Orange Knowledge Program for the international short courses on integrated pest management and food safety last June 2017 in the Netherlands. And another is on climate change adaptation in agriculture and natural resource management last April 2018 in Uganda. And for today's webinar episode, her talk will include part of her PhD research she's currently doing in New Zealand. She will be talking about microbes naturally found in the soil that can be used for pest management. She will discuss some examples of these microorganisms, mainly focusing on fungi. The title of her presentation is The Hidden Wonders of Soil, Nature's Solution for Pest Management. Without further ado, please welcome Ms. Michelle S. Guerrero. Let's give her a virtual round of applause. Ms. Guerrero?
Thank you so much, Ate Joy, for that very generous introduction. Can everyone hear me? Loud and clear?
Yes, loud and
Yes.
clear.
All right, thank you. Thank you so much once again, Ate Joy, for introducing me. Again, welcome. Isang magandang umaga po sa ating lahat, sa lahat ng nakikinig at sumusubaybay sa ating webinar series sa Zoom, YouTube, and Facebook. So magandang umaga po sa atin sa Pilipinas and good afternoon po this side. So in today's webinar, I will be talking about soil. So yes, hindi po tayo nagkakamali ng webinar series. This is still on pest management, but later we will know that soil is one of the source of microorganisms that we can use for pest management. So so as mentioned, so sa kasalukuyan, ako po ay nasa New Zealand. Last 2019, I got the opportunity to come here and do my PhD research. I've graduated my bachelor's and a master's degree in my home institute, my home university, my alma mater, the University of the Philippines, Los Baños, to which I am currently a university researcher, but now on study leave. And I'm working under the National Crop Protection Center, which brings you this webinar series. So last 2019, I received a New Zealand scholarships. So for those students in the audience, if you are interested in my journey on how I am able to receive this scholarship, I'm very much willing to share with you my experience. And because of this scholarship, I was able to take my PhD here in Massey University, New Zealand. So sa mga nagtatanong po kung ilang taon pa po ako, I started 2019 and the scholarship contract is 3.5 years. So hopefully on September 2022, I'll be able to graduate timely and give back to my home institute the learnings that I have learned from my degree. So just a recap of the first few episodes of the webinar series. Episode 1, scientists Mar and Sally Navasero discussed about several insect pests. So they discuss about onion armyworm and fall armyworm. And if you're able to watch that episode, more likely you've heard about Metarhizium relayi. And last Wednesday, my colleague Jenna Joy Apolinario, she shared about insect pest of mango. And mango sessid fly is one of the most problematic insect pests of mango, as she discussed last Wednesday. And interestingly, under BAFS, or the Bureau of Agriculture and Fisheries standard, there is only one organic microbials registered in our country in the Philippines. And interestingly, that's for sessid fly and seed borer in mango. So these are just the microorganisms that are very helpful in the pest management of different crops. So where can we find these microorganisms in nature? One of the places that we can see these microorganisms in nature is in the soil. So my topic for today is the hidden wonders of the soil, nature's solution for pest management. So as Ate Joy mentioned, this is part of the results that I will be showing you is part of the results that I have for my PhD research. So with me are my supervisors, Steve Trewick and Maria Minor from um, Massey University, and Dr. Travis Glare from the Bioprotection Research Center in Lincoln University. To start with, insect pest is one of the major threats to food security due to the damage and losses it inflicts to agriculture. And these threats are exacerbated by climate change and the loss of natural enemies, which controls pests in the natural ecosystem. Very true in the Philippines and here in New Zealand as well. Chemical control is one of the most popular control measure that farmers do to control insect pests and other pests in general. However, intensive use of chemical control poses risk to farmers, the environment, non-target organisms, and to consumers due to residues-related concerns. And intensive use of chemicals as well is also difficult to reconcile with the government, New Zealand government, for example, and the Philippine government, um, with our goal to be world leading in environmentally sustainable agriculture. That's why there is biological control as part of IPM or integrated pest management. And biological control offers alternative control measure which could be sustainable and environmentally friendly. So some of the groups of biological control agents are here. We can see in the screen, predators and parasitoids. As if you have watched episode 1 and 2 of the webinar, scientists Mar and Sally Navasero and Jenna introduced you to different examples of predators and parasitoids. But for this particular presentation, I'll be introducing you to the world of entomopathogens. So, as I've mentioned earlier, in the Philippines for now, we have one registered organic microbial. It is really hard to have a particular microbial in the market. As they say, it takes 5 to 7 years from discovery to product development and to offering that in the market for one particular microbials to be able for our farmers to be used. So this particular picture is one commercially available microbials for the farmers. So this one is against grass grub. But apart from that, here in New Zealand, there's also Bublast, which is the active ingredient is Beauveria bassiana, and Entokill and Entoblast. And the active ingredient for that is Lecanicilium. So what are entomopathogens? So entomopathogens are microorganisms that causes diseases in insects. Yes, ang mga insekto po ay nagkakasakit din. At ang nagdudulot ng mga sakit na ito ay ang mga entomopathogens. So ano po ba ang example ng entomopathogens? Some examples of entomopathogens are bacteria, fungi, nematodes, and viruses. So, iba't ibang microorganisms, iba't ibang infection route. So, for example, bacteria and viruses, they need to be ingested by the target pest for the effect to be seen against insect. But for fungi, for example, which is the subject of my research, they don't need to be ingested. They just need to be attached in the cuticle. And when favorable conditions are met, these fungi will grow in the insect and will continue its life cycle. So here in this picture, you'll see this one is a dead weta. So weta is an endemic insect here in New Zealand. And you can see the white spots here are the conidial growth. So from the conidia, it will attach to the insect body. And when conditions are favorable, it will germinate and penetrate within the insect body and ultimately causing insect death. So in this diagram, you'll be able to see the detailed mode of action of entomopathogenic fungi. So as I've mentioned, these microorganisms can be found in nature. So there are two sources of these microorganisms that can be used in pest management. One is through infected insects, as you can see in this picture. This is infected ambrosia beetle. And the subject of my research, and very new to me because I haven't done this type of research as well in the Philippines, is that the soil also inhabits microorganisms that we can use for pest management. So the distribution of different entomopathogenic fungi is well known, but more likely those distributions are from infected insects. That's why there's another um, component in nature that we can resort to be able to isolate these microorganisms. So as we know, the soil has complex interaction. It's not just the microorganisms, but soil inhabits different microarthropods as well, or other arthropods and other organisms. And the complexity of this soil microbial community, we would like to know what compose the diversity of soil, and we've done that using metagenomic approach. So later on, I'll be able to show you the detailed example of how we did the metagenomic approach of the soil microbiome. So apart from isolation-based approach, there's, as I've mentioned, there's a molecular approach in knowing what microorganisms are there in the soil. And that is through metagenomic approaches such as whole genome shotgun sequencing and amplicon sequencing. So yeah, the EPF products here in New Zealand are as follows: Entokill and Entoblast and Beauveria bassiana. Here we have Bugenic and Bublast. So both of these are in liquid form. And for us to be able to get to a particular product, selection of a virulent strain is very much necessary. So now I'm conducting efficacy tests. And apart from conidia as a propagule, I'm also trying to produce microsclerotia. So what are microsclerotia? Microsclerotia are it's one of another effective mass production protocol using liquid culture. So microsclerotia are fungal aggregates and are compact aggregates of hypha and are formed under unfavorable condition. So the usual practice that we do in the farmer's field is we get the conidia of the fungi and spray it directly to the crop. But most often, this method poses challenges such as UV degradation and other environmental challenges rendering its ineffectivity. For microsclerotia, these are often melanized, and there are certain endogenous reserves for use when favorable conditions return. So it's like an overwintering stage of the fungi. So what I have done, so as I've mentioned, soil is my main sample for my research. So I have collected numerous number of soil samples using a soil core. So this is a soil core. It's a 5x10 core. And my target habitats are forest, border, and pasture. So when I'm collecting soil samples, I'm also gathering data such as soil temperature and soil moisture. So there's a soil moisture meter that can be used in the field to know the optimum moisture of the soil, and that we will know that there is highly more likely that there's an EPF in that particular soil sample. So in the Philippines, there's one project doing entomopathogens on rice. And when I was doing this and I'm successful in doing my several techniques using soil samples, I have asked the university research associate that time, Janice Laquinta, if we could try to repeat this same setup, and we would be able to obtain entomopathogenic fungi. But maybe because of other parameters such as the soil is so moist, the insect baits are not able to grow in that particular example. So, yep, I'm conducting several soil collection in several places here in New Zealand, in Taranaki, Wanganui, and Manawatu. So all of these are in the North Island. So I'm here in Palmerston North. So that's in the North Island of New Zealand. So another picture of field collection. So apart from collecting soil samples across habitats, I'm also collecting soil samples across elevation, and we would like to know the diversity of entomopathogenic fungi across different elevation gradient. So this is what I'm mentioning earlier. So in my experience, it has been very encouraging that I'm able to obtain different isolates of entomopathogenic fungi just using this very, very simple approach, insect bait method. So in the beginning, I'm using Galleria melonelia, or greater wax moth, and Tenebrio molitor. So I thought initially that greater wax moth will be more ideal than mealworm as a bait because as we can see, the skin is so soft. So ideally, the fungi would want to go in to penetrate a softer cuticle rather than the mealworm. But in my results, um, I found out that the Nibrio molitor is a better insect bait. So for researchers out there who would want to try this method, I'm encouraging you to use mealworm for the insect bait method. So there's another method that I've tried in order to isolate the entomopathogenic fungi, and this is called direct plating in semi-selective media. It's a series of steps and a series of process. So for those interested to do this particular method, I'm willing to share with you the protocol. My email address will be flashed before I finish, so just take note of it. So, yep, and so this direct plating in semi-selective media, it will go through several soil dilutions. And from there, we will be able to obtain our beneficial microorganism. So for this method, we don't use the usual PDA that we use for growing fungi. Instead, we use a semi-selective media. So if you also want the recipe of that, just take note of my email and I'll be able to send you. Ayan. So yan po yung mga process in order to get EPF from direct plating method. All right. Yeah, so that's plating. So in order to arrive to a pure colony, it's not as easy as it looks. So there are challenges such as contamination upon contamination upon contamination of plates. So those who are working on fungal research would know this pain. So these are contaminated plates, and the other one you see on the right, is it right? Part of your screen is a pure colony. So identification of entomopathogens, specifically entomopathogenic entomopathogenic fungi, sorry, why is it nematode? Entomopathogenic fungi is hard such that you cannot rely just on the color. So I have also employed morphological and molecular approaches to identify my fungi. So there's one new extraction protocol that I've learned from here. So before, I'm using a kit-based approach in order to extract DNA. But there's one method using calyx approach, and this one is very rapid in terms of just few hours, and you'll be able to get your DNA. And this is just an example of the amplicon I have obtained using calyx approach. So yes, for those who again want to know about the extraction protocol using calyx-based approach, feel free to email me and I'll be able to. It's just that there's a lot of things and I can't be able to cover the protocol and all. That's why I'm telling that. So I have collected different soil sample. One I have subjected to insect bait method. The other one I have subjected to direct plating method. And the other one, I have done the modified Berlesi tool grain apparatus. So this machine does that, this piece of equipment does that. And so for that, I'm able to identify what soil microarthropods are there in a particular soil system that I have sampled. So sharing with you some results that I've got. So for the percent EPF infection, so remember I have forest, boundary, and pasture. It's always apparent that forest has the highest EPF infection, and this is just an example of the EPF that the infected mealworm that I have obtained. So this one, I've just put this in this slide just to, in this presentation, just to show you that not all insects with spores on it, like you can see in here, there's like a greenish colored spore. But if you notice, the body is not hardened. So if the body of the insect is not hardened, definitely it's not entomopathogenic fungi, although there are spore growth. And one thing as well is When I received the sample, I definitely know it's not entomopathogenic fungi because it's pungent, so mabaho. Pag yung insekto po ay may fungal growth nga, pero malambot at mabaho, malamang hindi po yun entomopathogenic fungi, more likely mga saprophytic fungi lamang po ang pumatay sa insektong ito. Ayan. So, yung ibang example naman po na yung ginawa ko using direct plating method. So here is the result. For forest, pasture, and boundary, it's still apparent that forest has the highest EPF per gram of soil. So the biodiversity of forest as a habitat is very much in terms of the abundance of EPF and other microorganisms as well, as you can see later in the metagenomic result of my data. So most of the counted entomopathogenic fungi were white, suggesting that it is Beauveria. So as I've mentioned earlier, it's really hard to identify the species of entomopathogenic fungi just by looking at it, because Beauveria, for example, it's white EPF as we sometimes refer to. But then, just showing you that I have done molecular identification as well, because it's really hard to determine the species using just usual morphological approach. So here in this slide, it has like 1, 2, 3 sets of pictures. And it's really hard to tell, right? What species is what. But if you're doing entomopathogenic fungal works for several times, you'll be able to notice the differences already. So for example, this one has, yes, it's white, but then um, it has pinkish glow, which we can see here. And this one is Beauveria malawiensis. And this one, this is harder because both are white. This one is white, this one is white. This one, I can easily say that it's not Beauveria bassiana, the common one. But then this one are both white. So it's really hard to tell other than using molecular approaches. So for those as well who's doing molecular identification works on entomopathogenic fungi, for example, it's best to use several molecular markers, such as we just not rely on ITS as a marker. So for this one, we need to use elongation factor as well and block primers. So this one is Beauveria pseudo bassiana, and this one is Beauveria bassiana. So sometimes it's really hard to tell just by looking at the plates. So as I have discussed earlier, I have done Berlacey extraction of my soil samples, and for the abundance of microarthropod, forest has the highest abundance as well. So sharing you as well the works I've done for the soil microbiome data. So for that, the other soil sample that I have collected, I have subjected that to DNA extraction. So I'm using this kit. This is called PowerMax Soil Kit from QIAGEN. And this is just the example of the soil DNA resolved through gel electrophoresis. And that soil DNA, we have sent it to third-party sequencing provider. And we've done shotgun sequencing and amplicon sequencing to determine the diversity of microorganisms that we have in our soil samples. And after that, there's the most challenging part of the research, which is doing the metagenomic analysis. So for my metagenomic analysis, I have used two platforms, Kaiju and Megan. So Kaiju is a program for, so if there are metagenomic researchers in the audience, so these are some of the useful platforms that we can use for metagenomic analysis. So Kaiju, it's a program sensitive for taxonomic application of high throughput sequencing reads from metagenomic whole genome sequence data. And Megan, it optimized analysis of large metagenomic data sets. So these are just results of my Kaiju analysis. So that one, actually it's just 10 grams of soil that was extracted. And in that 10 grams of soil, usually it has more bacteria than my target fungi. So, but still, Kaiju as a program, it was able to give me 31 Bouveria and 205 reeds of Metarhizium for forest and 27 Bouveria for pasture and 152 reeds for Metarhizium. So for my soil DNA as well, I was able to know that there are other Bouveria species present in the soil sample. So there's Bouveria bassiana, Bouveria caledonica, and different species of Metarhizium. So doing the metagenomic analysis and soil DNA extraction, it gives us idea that we could go back to that particular site, and if we're not able to isolate the particular species using plating and other methods, then we could possibly try again and be able to obtain other species. So this is just the pipeline used for the metagenomic approach. So this is the result of Megan. It's just giving you comparative result of other OTUs seen in the data. Yep, just showing you some examples for the benefit of those doing genomic or metagenomic researches. Yan. So, nalalapit na po tayo sa pagtatapos ng aking talk. Ayan. So, sa ngayon po, medyo masalimuot, I'm doing a lot of efficacy trials. So my target insect pests are giant willow aphid and grass grubs. So because here in New Zealand, they have vast pastures and milk is, it's yun, mura lang yung milk po dito kasi ang daming production. And one of the number one insect pest for pasture are grass grubs. And the number one pest for willows, so this is the pest of willow tree, are giant willow aphid. So I've been doing efficacy test. So for my laboratory efficacy test, I'm doing, there are two methods that can be used. One is through the use of airbrush technique. So airbrush technique is better than the usual sprayer because hindi nalulunod yung mga insekto. So you have the possibility na mamamatay sila using through EPF. And this one is Potter's Tower. So very efficient equipment as well for efficacy trials for entomopathogenic fungi. I'm doing efficacy test on grass grubs as well. So a lot of grubs has been collected in the field and being tested. For now as well, as what I've mentioned, apart from testing my isolates using conidia, I'm also producing microsclerotia. So microsclerotia is, I can't say that it's better, but it's an effective propagule in unfavorable weather conditions. So I hope this microsclerotia will be, I hope to formulate it into granules and be able to test it in the field and see its effectivity. Because these microsclerotia, they can survive in unfavorable condition, and they will, so it's better in a sense that they can survive in unfavorable condition, and when the insects are there and the proper humidity are there, they would just be able to infect the insect pests. So ayun po, kung may mga if you want the recipe of the semi-selective media or the protocol for Kilex extraction and other things na hindi ko po na-share dito because of time limitation, you can email me at this following email address. So, nawa po ay marami po kayong natutunan. I hope you learned something. Just showing you some of my pictures of my journey here in New Zealand. And I would like to acknowledge National Crop Protection Center and CAFS and UPLB for allowing me to do my study leave and take my PhD. I'm also thanking my wonderful supervisors who helped me with my research here in New Zealand. So ayun po, maraming salamat at nasa ano ba ako? Within the time limit? Anyway, yun po. Thank you very much everyone for listening, and I hope you'd learn something from my talk. And in these trying times, trying and challenging times, I hope that we'll be able to foster collaboration and work together for the crop protection, for the advancement of crop protection in the Philippines and in other parts of the world. Thank you so much. Maraming salamat.
Thank you very much, Ms. Mitch, for that very interesting and informative talk on soil microbes, entomopathogenic fungi, or EPF. Okay? Thank you, Ms. Mitch.
All right.
Okay. So,
did I stop my share screen already? All good?
Yeah, thank you. Yeah, all good.
Okay.
Okay. Napaka-interesting po talaga nung topic ni Ms. Mitch. Ano po? And we just like to acknowledge yung hindi po natin nabati kanina. From ang pinakamadami pong participants, attendees ay galing sa Benguet, sa Nueva Ecija, La Trinidad. Magandang umaga po sa ating lahat. And we also have an attendee from South Africa. Hi, Chima. And then we also have attendees from Palawan, from Thailand. Good morning. From Batangas, and of course from Los Baños, from Maguindanao, and from sino pa po ba hindi natin nabati? From Apayao, General Santos City, Santo Tomas, Batangas, Zamboanga del Norte. Parami po tayo. Okay, sige po. Salamat. Salamat po sa inyong pag-attend. Paalaala po na kung nais po ninyong makatanggap ng e-certificate, mangyaring punan po natin ang evaluation form na kasalukuyang isa-share po sa atin ng ating team, na isa-share din po sa mga chat boxes sa Zoom, FB at YouTube. Naririto rin po ang QR code at link para sa pag-fill out ng evaluation form. Kami po ay nagpapasalamat sa inyo dahil ito po ay makatulong upang mas lalo pa naming mapagbuti ang paghahandog sa inyo ng webinar na katulad nito. Maraming salamat po. At habang nagbabahagi ang ating speaker, ay mini-monitor ng ating webinar team ang inyong mga katanungan. At upang matulungan po tayo upang ipahayag ang ating mga questions, narito po ang ating moderator for today's webinar. May I call on Ms. Maureen Ceres de Rojas, isang entomologist and university researcher mula din po sa NCPC. Ms. Maureen?
Thank you, John. Thank you, Miss Michelle, for that wonderful talk. Ayan. Wait na. Ayan, siksik na naman po ang ating impormasyon sa ating webinar for today. I too believe in the power of soil to heal. I think even in the medical field, they found solution from the soil, ano? With the importance of soil in agriculture, pest management, and in the medical field, and in many other fields, huwag po natin kakalimutan na i-conserve ang soil, okay? So, Miss Michelle, ready ka na ba for the questions?
Yes po.
Yeah?
Very
Yeah.
pretty.
Okay. So, the first question is from Sir Dave General. Hi po, Sir Dave. I think from from MNH. Ang platform po na ito, na kanyang pinagtanungan ay from FB. Ang question po, good morning po. Any ants from the modified toll grid extraction? I am an ant specialist at UPLB MNH and I'd like to examine any ants that you collected.
Yeah, I have collected ants from my Burlesy extraction, but yeah. So if the question if I have collected, yes, I have collected using the Burlesy extraction. Did I answer it correctly? Yeah, but
Yes,
yeah,
are you willing to share? Pwede
yeah,
bang
yun
iuwi?
pwede bang iuwi? Yun na. Pwede sigurong iuwi kasi patay na, pero I'll look into it po, sir. I'll look into it.
Okay, thank you, sir. Thank you, Miss Michelle. This is a comment from Ma'am Au Pinon. Ma'am Au, we are very grateful at suking-suki po kayo sa ating webinar. Ang sabi ni Ma'am Au from Zoom, ano? Thanks, Ms. Guerrero, for new information about some helpful fungi you have done there. Ayan.
Thank you, ma'am.
From Ma'am Marcela Balbon from Zoom. Ang sabi niya, thank you, Miss Michelle. I love the way you say fungi. Tama ba?
Oh,
Ano
yeah.
nga ba?
Before po kasi, nung pumunta ako dito, I'm trained as, ngayon hindi ko na mabalik. Sa Pilipinas kasi, fungi. Pag nandito po, kagaya ng mga iba't ibang termino ng English, yung tomato nila dito, tomato. So, ang fungi nila dito ay fungi. So, nasanay na po ako na, ayun, I just got used to using fungi instead of fungi.
Okay. So,
Ang virus mo din, virus.
Hindi naman po. Virus pa din, sir. Kung ang virus
From,
daw yung virus.
Joke lang.
Virus pa din.
from
Yun lang, sir,
Mr.
sa ibang words.
Roderick Granada, sa Zoom, naman din po. Sabi niya, Kia ora, ma'am. And I would like to ask if how hard to produce microsclerotia? Is it also quantifiable like the common conidial structures?
Yep, quantifiable din po siya. I'm still in the process of optimizing the protocol. Hindi po siya ganun kadali because you just need a liquid media. Well, just to start with, you need a liquid media for the production. Of course, you need your conidia, liquid media, and you need to have, of course, you incubate it, shake it, and all, just the usual fungi. But there are certain temperatures and shaking time and all. So you need to account that factors as well. So kung Mahirap, hindi po siya mahirap, but kailangan lang i-optimize po yung protocol. Kasi just to clarify as well, hindi po lahat ng entomopathogenic fungal species are able to produce microsclerotia. So Beauveria bassiana, it was found to produce microsclerotia, but Metarhizium anisopliae, yes, but other Metarhizium species, they are not able to produce microsclerotia. So madali po dun sa mga species na kayang mag-produce ng MS, ng microsclerotia. At kung yung species po ay hindi talaga nakaka-produce ng microsclerotia, then hindi po talaga makaka-produce. Pero kaya po natin gawin.
Okay. Thanks, Miss Michelle. Another question. This is from Ma'am Karen Alviar from UPLB. Hello, ma'am. Sa iWeb po ito si ma'am. Ano? Ang platform niya po ay Zoom. So, ang tanong niya, her question is, can kaiju be used for other applications on taxonomic classifications other than metagenomics?
The answer is no po, kasi kaiju is just for it's for metagenomic and metatranscriptomic. So if the question is if there are other platforms other sequencing methods that you can use kaiju for, you can use it using metagenomics and metatranscriptomics. Yung kaiju kasi it's it analyzes environmental samples, so soil samples, any extraction that you have with you don't know what's inside. So kaiju would be able to resolve up until species level, which is kind of encouraging. Yep. Pero apart from metagenomic data, hindi po. Even my amplicon sequencing, so metagenomic sequencing po kasi, whole genome metagenomic shotgun sequencing, ito yung, you have your environmental DNA. For example, my one is soil DNA, and I did whole genome shotgun sequencing. So, ibig sabihin, it sequenced all of the organism, possibly all of the, or most of the organism, using different gene region. So for the whole genome shotgun sequencing, I was able to use kaiju, but for my amplicon sequencing, which is the same soil DNA, but targeting one gene region, which is ITS, I used ITS for my soil DNA data. For my soil DNA, hindi po applicable ang kaiju doon. So, yun po.
Okay. Another question from Mr. Francisco Soriano via Zoom. His question is, are there biosafety and biosecurity issues related with the use of EPF as a biocontrol agent?
Sorry, Miss Mao. Sorry,
Okay.
I was just, yeah.
Okay. Sabi ni Mr. Soriano, are there biosafety and biosecurity issues related with the use of EPF as a biocontrol agent?
Wala akong, not that I know of, sir, ano po, here in New Zealand as well. But for sure, if that entomopathogenic species is a new species in a certain country, then biosafety protocol should apply. Yun po.
Okay, next question from Mr. Jeffrey P. Villaver. Via Zoom din po ito, no? Fall armyworm is one of the most destructive pests in corn today. May I know if the researcher has tried or planning to test the efficacy of entomopathogen in FAO?
Meron pong other colleagues like scientist Mar Navasero, who discussed about fall armyworm last time. He already discussed about epizootics of Metarhizium relayi. So more likely, the group of scientists Mar Navasero will be able to address FAO. So currently po kasi wala po ako sa Pilipinas, so I'm not able to do researches on FAO in the Philippines. Yun po.
Okay, another question from, oh no, this is a comment from Ma'am Maria Divina Gracia de los Trinos via Zoom. And sabi po niya, hello Michelle, nice to meet you here. Bago pa lang ako sa farming. I have nothing to ask for now. Maybe sa pagkikita natin. Thank you so much.
Yes
Ayan. So
po, next year po.
na-appreciate nila, no?
Ayun po.
So another question from Stephen Arthur via Zoom. Is
Oh
it
my.
possible to collect samples from cultivated fields to know how much the use of pesticides have affected the population of these beneficial microorganisms?
Yes po. You can do that, Stephen, no, Arthur. May mga sorry, should be in English. There are studies in literature doing that, comparison of soil, isolation of EPF from soil with, heavy chemical sprays from those with most farms without sprays. So yeah, it's being done. And you'd notice that in some literatures, they say that it affects, definitely. So if for example, in my samples, pasture has lesser diversity and abundance of entomopathogenic fungi than forest. So, and with my Berlacea tulgrin extraction, I found out that in pasture, there's very, very less diversity of soil microarthropods. So, because EPF depends on insect host, so it follows. So, yep, you can do that definitely to check. And you can do that for research purposes as well.
Okay, another question from Ma'am Myra Domingo via Zoom ulit. No? Sabi niya, hello po ma'am. Just like you said po that we cannot request for some protocol, can we also request list of primers that you use to identify your community samples before sequencing? Thank you po.
Community, well, I can send po I can share primers used for molecular identification of entomopathogenic fungi, but it doesn't apply for the whole genome sequencing. But definitely for the elongation factor, internal transcribe spacer, and block primers, yes po, I can definitely share. So feel free to email me po.
So ayan, mukhang nahihiya silang magbigay ng questions. Can you share ulit your email address if they are also have questions pa?
Okay, so in the chat box or just...
Yes. Yes.
Okay, sige po.
Okay.
Ayun po, ito po ang aking email address. May natutunan po ba kayo?
Napaka siksik naman sa information. Talaga namang marami kaming natutunan, Miss Michelle. So, you can contact Miss Michelle at msguerrero1@up.edu.ph or at m.guerrero@masi.ac.nz. Oh, there is another question from Mr. Renson Marqueses. Ano, may pahabol po tayo. Sabi niya, Ma'am, good day. Ma-minimize po ba ang fermented pesticide ang mga pest? Tama ba ang aking ano?
Fermented pesticide. Medyo wala pa po akong pag-aaral. I'm focus on entomopathogens, but maybe ang ibig sabihin kaya ni sir ay yung mga fermented pesticides like yung mga from EM and all. Baka.
Turo po,
Pero
yung mga
wala po akong dati po sa East West, ginamit po namin yan. Before I worked as a junior technology transfer specialist at East West Seed Company. And we've used effective microorganism for pest management. But yes, it helps. It helps if yun po yung tanong. Nakakatulong po ang effective microorganism, fermented microorganism. But as a researcher, wala pa po akong pag-aaral tungkol po doon.
Okay. So mukhang on time po tayo ngayon, ano? Again, i-share po natin yung email address ni Ms. Michelle Guerrero sa chat box. If you have other questions, you can email her. And also, if you need other, if you need other services from NCPC, you can also email ncpc.uplb.edu.ph. I am bringing you back to Ms. Mary Joy Mendoza. Ms. Joy?
Thank you very much, Ms. Maureen and Ms. Mitch, for that wonderful discussion. We're just on time. So, muli, reminder po lamang na pakisagutan po ang ating evaluation form na nakashare sa ating chat box sa Zoom, FB, at YouTube. Ito po yung QR code at saka yung link po. Maraming salamat po. Ayan, so malapit na po tayong matapos. And for closing remarks, may I call on our Deputy Director, Ms. April Alviar. Ms. April?
Pagpalang umaga po sa ating lahat. Isa na naman pong webinar ang inihandog ng National Crop Protection Center bilang bahagi ng selebrasyon ng aming ika-65 anibersaryo. Marami pong salamat sa ating resource person sa umagang ito, si Ms. Michelle Guerrero, na bagamat nasa New Zealand upang mag-aral, ay hindi naging hadlang ito upang ibahagi ang kanyang kaalaman tungkol sa soil parasitic microbes at kung paano po ito gagamitin sa pest management. Puso din po kami nagpapasalamat sa inyong lahat na patuloy na sumusuporta sa aming webinar, sa inyong pakikinig at naway ang inyong kaalaman natutunan sa umagang ito ay inyo pong magamit sa tamang pamamaraan. Papasalamat din po kami sa lahat ng bumubuo po ng NCPC webinar team, NCPC Extension Team, at CAPS Extension Team sa patuloy na pagtsatsaga upang makapaghatid po sa inyo ng makabuluhang webinar. Muli po namin kayo iniimbitahan sa May 26, 2021 sa huling bahagi po ng aming webinar series na ihahandog po ng aming isa pang researcher, si Dr. Marita Pinili. Kita-kits po tayo sa susunod na webinar. Muli, isang magandang umaga po sa ating lahat.
Maraming salamat, Ms. April. Dito na po natatapos ang ating third webinar episode. At gusto din po namin kayong ibitahan, gaya po na nasabi ni Ms. April, sa susunod nating webinar. Ang title po ay Crops Diseases Beyond Boundaries. Know the Transboundary Diseases of Trees, Cassava, Cacao, Corn, ni Dr. Marita Pinili, Mr. Freddy Webb Signabon, and yours truly sa May 26, 2021 sa parehong oras po, alas 9 ng umaga. Okay, so iniimbitahan din po namin kayo na tunghayan din sa NCPC FB page ang ating Mobile Photography Challenge. Inaanyayahan po namin kayong bumoto sa mga photos na nakapost doon. May online voting po tayong nagaganap at ito ay hanggang bukas na lamang, May 15, 12 noon. Huwag pong mahiyang i-like, subscribe, follow, at share ang ating FB page at ang ating YouTube page para po hindi tayo mahuli sa mga kaganapan. Muli, ako po si Mary Joy Mendoza. In behalf of the webinar series team, we would like to thank everyone for attending and participating. Maraming salamat po. Keep safe and God bless everyone. Mabuhay po kayo. Maaari po ba tayong magkaroon ng group photo shoot? Maaari po natin open ang ating video. Take it away, Mau. Okay.
Lagi po sa ganitong group photo, hindi ako nakatingin sa camera. Wait lang po. Dali lang po, no? Isa pa. Wait lang. Ano ba yan? Okay, 1, 2, 3. Okay. Yan. Thank you po ulit.